Okay, so in part 4, 5, and 6 of this chapter, we're going to be talking about situations where traits that we're studying are going to violate Mendel's two laws. And so these are all going to be exceptions to Mendel's principles. And so we're going to start off by talking about something called incomplete dominance. Okay? And so in the picture here to my left, our left, I've got a picture of a flowering plant called the snapdragon. And so snapdragons are a key textbook example of incomplete dominance, one of them. I'll show you another one on the next slide. And so what is incomplete dominance? So we've heard now about dominant and recessive alleles, right? And so if a trait follows Mendelian rules, then the dominant allele is always expressed, whether it's a homozygous dominant or heterozygous individual. And the only way to get the recessive phenotype is in the case of a homozygous recessive organism. In incomplete dominance, it's a little different. And so in incomplete dominance, the heterozygote is actually going to express an intermediate phenotype. And so there's not really true dominance and recessive here. Instead, what we get in the heterozygote, again, is an intermediate phenotype, which looks kind of like a blending of the two parents. And so this phenotypic ratio of 1 to 2 to 1 is going to be really important here. So if incomplete dominance is at play, and we have a cross between two heterozygote organisms, we will end up with a 1 to 2 to 1 phenotypic ratio, as well as genotypic ratio. And so let me show you an example of this. We'll come back to snapdragons in just a second. Okay, so these are 4 o'clock flowers. Again, another textbook kind of example. And so we're going to start off here at the top, just like Mendel did in these studies. What we start off with is true breeding pure lines. And right here, we have a red flower producing 4 o'clock plant. And over here on the left, our second parent in the parental generation is going to be a true breeding pure white flower producing plant. So those are the phenotypes. Now, genotypes for incomplete dominance are going to be different than what we've seen so far. And so what we're going to do for red flower producing plants, we're going to have 2 alleles. So this is the R allele. And for red, we're going to call it R1. And if we have a red flower, it's going to have 2 of the same alleles. So R1, R1 would be the genotype for a red flower producing plant. For a white flower producing plant, we're going to use R2, R2. So again, very different. We're not going to use just a capital letter and a lowercase letter, right? Because we don't have true dominant and recessive. So instead, we're going to use this superscript to the right of the letter. Okay, so again, one allele is R1, the second allele is R2. For a homozygous red flower producing plant, the genotype would be R1, R1. For a homozygous white flower producing plant, the genotype would be R2, R2. Now, if we cross these, you can set up your Punnett square if you need to. It'll just be a one-boxed square. So the red flower producing plant, all of its gametes will contain R1. And for the white flower producing plant, all of its gametes will contain R2. And so the product of this initial parental cross will be the F1, or the first filial generation. And all of the flowers here will have the genotype R1, R2, right? So the R1 allele came from the red flower producing plant. The R2 allele came from the white flower producing plant. Look at the phenotype. Okay, so again, if this was true dominance, then this F1 generation would look like one of the two parents. It doesn't. It looks like an intermediate. So it's not red and it's not white, but it's pink. Okay, so again, this is incomplete dominance. If we take this out to the F2 generation, which of course we do, okay, so, where's my pointer? Here we go. So we're going to cross 2 pink plants, so 2 heterozygote plants. So we'll put the gametes for one parent here on the left, and for the second parent up here on the top of the Punnett square. And so in both cases, the pink flowered plants are going to produce 50% of their gametes containing the R1 allele, and the other 50% of the gametes will have the R2 allele. So half and half. Okay, so here's R1 and R2 from one parent, and the gametes of the other parent again are across the top. So here's R1 and R2. Remember, inside of the Punnett square is the result of fertilization. So here R1 and R1 end up in the same cell, and we end up with offspring that are all R1, R1. These would be red flower producing plants. Here we have this gamete being fertilized by this gamete, and so we end up with R1, R2, pink. Okay, same thing over here, this gamete and this gamete, R1, R2. And lastly, in this box here, it would be this gamete and this gamete. So again, R2, R2. So for the phenotypic ratio, we have 1 red, 2 pink, and 1 white. That's the 1 to 2 to 1 ratio. And so again, this is going to be the result of the crossing of 2 heterozygotes if incomplete dominance is at play. There are other examples, and you'll see other ones when we look at genetics problems, but just to remind you back to the snapdragons. So this is the same picture that was on that first slide. And the same thing here, if we take 2 heterozygotes that are both pink flower producing plants, we will end up with that same ratio of 1 to 2 to 1, phenotypic and genotypic ratio in the offspring. So 1 red to 2 pink to 1 white. It's the same phenotype here for this other flower. In snapdragons, we use different representation here for alleles. In snapdragons, we use CR and CW, so color red, color white. So a red flower producing snapdragon plant would be CR, CR. A white flowered plant would be CW, CW. And a pink flowered plant would be CR, CW. Okay, so again, when we go to do genetics problems, you know, you can kind of pick whatever allele you want to pick. And again, there are alleles that people do generally use. And so again, if you have 4 o'clock flowers or snapdragons, you want to make sure that you use what, you know, the terminology that we know. You know, the same thing, you know, in lab, we're going to be using, you know, the letter R for color and the letter S for kernel shape. So we kind of want to stick to those the best we can. So incomplete dominance was our first example of a non-Mendelian inheritance pattern. Our second one here is codominance. And so codominance is actually a variation of incomplete dominance. And so in the case of codominance, both alleles for a given trait are expressed simultaneously in the heterozygote. You don't get a blended phenotype, but instead you see both, in this case, colors. Okay? And I'm going to give you another example where it's not colors. So let me just show you these 2 pictures. So on the right first, this is a speckled chicken. And so if you look at the colors of the feathers, they are brown and brown with speckles. Okay, so there's a brown allele and there's a speckle allele. Sorry, a brown allele and a white allele, and they both get expressed. So you end up with brown speckled feathers. Okay. So it's not an intermediate phenotype, but instead both are expressed simultaneously. Here in this other picture, we have a roan cow. And so again, you have 2 different alleles. One codes for brown and one codes for white. They're going to be both expressed simultaneously, but again, not blended. So, you know, we don't end up with lighter brown cows. Instead, we end up with cows that are brown and white. Okay, so their fur will be both colors. Okay, so again, codominance, both alleles get expressed simultaneously. Here's a really good example of codominance. This is one of my favorite examples. So you'll have lots of genetics questions on this. Blood type. Okay, and we're going to keep it simple. We're not going to actually talk about the Rh factor, but if you know about that, that adds another layer of complexity here. We're just going to talk about the 4 blood types. blood type A, B, AB, and O. Okay, and so I want to quickly go over phenotype and genotype. So if you are an individual who has type A blood, your phenotype would be type A. Your genotype, so what alleles you've inherited and are expressing, look like this. And so here is one potential genotype that gives you type A. So you could be IA, or you could also be IA little I. Okay, so you could have inherited either 2 of the IA alleles or 1 IA allele and 1 little I. What does the I stand for? It stands for immunoglobulin, which is a surface marker on your red blood cells. And on the next slide, I'll actually show you a picture of this. Okay, so if you are type A, you have one of 2 genotypes. The same thing for type B. So if you have type B blood, you can either be IB, which means you inherited 2 of the same IB alleles, or you might be IB little I. Okay, 2 different alleles here. Here is the codominant phenotype and genotype. So if you are type AB blood, that means that you inherited 1 IA allele and 1 IB allele. And both of these alleles are expressed at the same time. And so therefore, the surface of your red blood cells have both the A marker and the B marker. And again, I'm going to show you a picture of that in just a second. Lastly, there is the phenotype of type O blood. People who have type O blood have inherited 2 recessive alleles, little I, and people with type O blood actually don't have any surface markers, surface antigens. So let me again show you a picture of this. This is a nice little chart here. Okay, myself way over there. Let's ignore the question for just a second and let's just look here. So again, I give you alleles. So there are 3 alleles that exist in the human population for blood type. So there is the IA allele, the IB allele, and the little I. Okay, and what do these code for, these alleles? So the IA allele codes for the surface carbohydrate, which in this case we're drawing like a little triangle. It is the A surface carbohydrate. People with with the IB allele, I should say, will express this B carbohydrate. And so again, in this figure, I'm showing just a little round yellow circle. If you inherit the little I allele, this is considered to be recessive. And if we look at what carbohydrate is produced from this allele, there is none. Okay, so let's look at the potential, again, genotypes and phenotypes. So we said if you're type A, you are either IA or IA little I. And so the IA allele is going to be expressed even in this heterozygote condition. So if we were to take a look at your red blood cells would be, kind of coated by this surface antigen that again looks like a triangle, and it's a carbohydrate molecule. Okay, so we would find this surface antigen, which again we call A, on the surface of your red blood cells. If you're type B, right, your genotypes will be IB or IB little I, and your surface of your red blood cells are going to look very different from somebody with type A blood. You would have these little circles. So the carbohydrates on the surface of your red blood cells are different. If you have type B blood and you received blood from somebody who was type A, your immune system would recognize these surface antigens as being foreign and would actually destroy those red blood cells. Okay, so somebody with type B blood can't get type A, and somebody with type A blood cannot accept blood from somebody who's type B. Okay, let's go to AB. So if you have type AB blood, there's only one possible genotype that you can have, and it's IB. So you inherited one of each of those alleles. And so the surface of your red blood cells is coated with both the A and B carbohydrate. And so again, this is the instance of codominance. This is the codominant genotype and phenotype. Both of those alleles are expressed simultaneously, and so you end up with both surface carbohydrates. Okay? In the last case here, somebody with type O blood, you inherit 2 little I alleles. And if we were to look at the surface of your red blood cells, we would not find any blood type carbohydrates. Okay, we'll quickly just kind of talk a little bit more about, what kind of blood you can get. So if you are type O, you can only get type O blood. If you were to receive AB, B, or A, your immune system would see those surface markers as foreign, and again, would not accept. That blood would be rejected. If you are type AB, you can get blood from anybody. You are considered to be the universal acceptor. You can get type AB blood just fine. You can get type A blood because you have A surface carbohydrates. You can get type B blood because you already have those as well. And you can get type O. And so you can get blood from anybody. Again, I am leaving out the Rh factor. So if you know a little bit about that, I am simplifying it a bit. If you are type B, you can get blood from somebody who is type B or O, and that's it. If you are type A, you can get blood from somebody who's type A or O, and that's it. You can't get blood from B or AB because of that B surface carbohydrate. If you're type O, again, you can only get type O blood, but you can give blood to anyone. And so if you have type O blood, you are the universal donor. Okay? So again, it's the type AB genotype and phenotype that is our example here of codominance. Both are expressed simultaneously. Go on to the last example here. I want to quickly do a cross with you. You'll have to do this in lab and in lecture, and so want to make sure that you can do that. And so let me grab my pen here. We'll do black. Okay, so I'm going to cross an individual who is type AB. So here is the genotype IA, IB, with an individual that is also type AB. So IA, IB. And so again, both of these individuals are examples of codominance. They have both alleles, both would be expressed simultaneously. Their red blood cells would look like this over in this picture. Okay, and we would call them heterozygotes. And so I'm going to set up a Punnett square. And so we're just looking at one trait here, right? Just blood type. And so it is a nice, simple monohybrid. Okay, trying my best to make a straight box here using my mouse. Okay, so I'm going to put this parent's gametes over here on the of the box, and I will put this parent up here on the top, across the top of the box. And so just like usual in a monohybrid cross, these two alleles, so the IA and the IB allele, are going to segregate or separate from each other during meiosis, and they're going to end up in different gametes. So this parent's gametes, half of them, will be IA, and I actually, when you type a capital letter I, it looks like that, but I actually usually make it look like this. Okay, the other half of this parent's gametes will be IB. Okay, the same thing across the top. It's the same exact genotype. So half of the gametes from that parent will be IA, and the other half will be IB. Okay. So let's see, let me grab a different color here. So inside my box is here of my Punnett square, I'm going to show the product, right? So I'm going to show the offspring. So if IA and IA come together, I'm going to end up with offspring that are IA, IA. And if I move from left to right, go to my next box over, and there I've got, oop, I'm writing that wrong, hold on. Let me grab my eraser. Jumping to the next box too quickly. Okay. So this will be IA, IA, IA. caught my mistake there. Okay, now over to this box. This was going to be IA, IB. IA. Again, I usually like to put the top and bottom up, but for time's sake, I'll just leave it off. Those are capital letter I's, IA, IB. Down here, this is also going to be IA. Zygous. Those terms should be really familiar by now. And my last box here is going to be homozygous, IB, IB. And so we have to be careful with our genetics problems. We have to make sure that we actually answer the question. And so it said, if two heterozygous type AB individuals mate, what would be the phenotypic ratio of the offspring? And so look, so I've got a 1 to 2 to 1 ratio, just like I saw in incomplete dominance. So codominance, again, is a variation of incomplete dominance. So again, 1 IAIA, 2 heterozygotes, and then 1 IBIB. So there's that 1 to 2 to 1 phenotypic ratio. And again, we'll get lots of practice with blood type questions. Okay, the last slide here. Let me go back to my pointer. Okay, the last scenario, again, which violates Mendel's rules, is going to be multiple alleles. And so, you know, we've been talking about traits that kind of differ in this either or fashion until we got to blood type. So, you know, when we looked at, you know, flower color, you know, 2 possible alleles, okay, either the, you know, if we're talking about 4 o'clock flowers, either the red allele or the white allele. If we're talking about snapdragons, either the red allele or the white allele. You know, in lab, looking at corn kernel color, either the purple or the yellow allele. So that's, we've only been talking about 2 alleles really until we got to blood type. When we got to blood type, we said actually there's 3 alleles for blood type. There's IA, IB, and little I. So that's already one example of multiple alleles that we've seen. And that holds true for many traits. So usually there will be more than 2 potential alleles for a given trait that exists in a population. And organisms in the population like us, or this bunny pictured here, are only going to inherit 2 alleles out of that pool of alleles that exists because we're diploid. And so again, blood type was a great example, right? There were 3 alleles and individuals inherit 2 of those 3 alleles. The next example that I'm going to give you on the next slide is going to deal with coat color in rabbits. And so here we go. There are 4 alleles, and those are listed here at the top. There are 4 alleles that exist for coat colors. There's the capital C allele, there is the CCH allele, the CH allele, and the little c allele. Okay, and so for genotypes, this slide actually only gives you 4. So big C would give you brown fur. CCH would give you chinchilla fur, which looks like this bunny right here. So black-tipped, kind of grayish white fur. Himalayan is CHCH. This is white fur with black paws, nose, ears, and tail. And then little c is the albino genotype, which gives you the phenotype of white fur. Okay, I want to also mention to you that there's a dominance hierarchy. So what if we had a bunny that was C, big C, CCH? It would still be brown. So if this big C is with any of these other alleles, so big C, CH, big C, little c, you're always going to get a brown bunny. The second in the dominance hierarchy is the CCH allele. So CCH, CH would be chinchilla, CCH, C would be chinchilla. So we say that the capital letter C is dominant to the CCH allele, but the CCH allele is dominant to the CH and the C. So there's this hierarchy, and right in that order. And so the only way to get an albino white rabbit is if you have two little c alleles. If any other allele is present, it won't be an albino rabbit. Okay, so again, for most genes, more than two alleles exist in a population, and usually those alleles will exhibit some type of a dominance hierarchy. That's it for this fourth part of the chapter. The next part of the chapter will continue with traits that don't necessarily follow Mendel's rules.